Okay, hello again, everybody. Sorry for the technical problems yesterday. I'm not exactly sure what the problem is here, but I think sometimes our Wi-Fi is clamped a bit, and so we'll take that in consideration for future meetings like this. I'm sad that I don't get to present this where people could just ask questions. So I have included my email address here for any interested parties if they want to contact me to ask any questions about what I'm talking about today, or any questions about research in general or anything. So please feel free to do that if you would like. And as I mentioned yesterday, I think I got that far. I'm going to talk about mechanisms of breathing dysfunction during seizures. Yes, and I wanted to bring up one more thing before I go through the slides. I don't talk about SCNA or NAV 1.6 directly in this talk. I don't work on the channel directly, or sometimes I have in the lab done some like cellular recordings and things like that, but that's not mostly what I do. So it should just be noted, and I don't bring it up in detail on the slides, but all of the mouse models that I use they're SCNA mutant mice, and so that's why they have seizures and whatnot. So everything I'm going to show you today are using these mouse models that Dr. Miriam Meisler created at the University of Michigan. But I'm not going to talk about the channel per se, just so we're aware of that. Okay, so the significance of this work, this is always a very critical thing when in research, is defining the significance. And so the purpose of looking at breathing during seizures is related to SUDEP, or sudden unexpected death in epilepsy. So SUDEP is unexpected. It's also unexplained. And so over the past decade or so, there's been a big push to understand what is or may be happening with this fatal complication of epilepsy. There's still a lot to understand. Is the short of that, but it's a major focus area. The NINDS, the neurological disorders at NIH, lists SUDEP is one of the important areas to understand going forward. And so it's a highly significant topic. Right now, it's thought that SUDEP occurs because of failure of breathing or cardiac function, or both. There's a big push, at least in the circles that I am involved in, that the breathing is a big one. And mostly I'm not talking about breathing today, because at least in this mouse model, that seems to be the system that's really compromised during seizures. I guess I should say that the general consensus is that SUDEP occurs immediately after a convulsive seizure. That's the idea. Although most are unwitnessed, and we really just don't know all of what's going on. And so this is a scenario where using a mouse model is extremely pertinent, because we can record them, we can observe them, we can do this without any treatment. So the mice that I'm going to show you are not receiving anticonvulsant drugs. In fact, they have to live on their own. They undergo surgeries so that I can record their brain and ECG and things like that. They have to live alone to do the breathing measurements that I do. So we're pushing them sort of in that direction. And then finally, there's just power in numbers. Mice breed like mice, and they don't live very long normally anyway. So we can have many, many mice, and even if whatever we're looking at isn't that common, we can identify it and record it and try to understand it. So that's the purpose of this work. So I wanted to cover the significance first, but then a brief outline, just so you know where I'm going in this presentation. I'll talk a couple slides about introduction to the systems that I study, and then how we do it. Then we'll talk about the different types of seizures these mice have, notably tonic and clonic seizures. I'll show a video of a tonic seizure, because that's sort of the important one for what I've observed, which will be an example of tonic phase apnea If you search this term, you may or may not find much about it. It's a term that I'm kind of inventing, I suppose. Apnea just means no breathing, literally from Greek. And tonic phase because the apnea that I see in these mice occurs during the tonic phase of seizures. And I think that it's very significant. Like I say, I think breathing is the important, the failure of breathing is the important aspect in these mice. And so I'm going to show you that this apnea, if we can prevent it with this artificial ventilation, we can prevent SUDEP in these mice in a certain model that we have. I'm going to talk about these different types of seizures and how they contribute to SUDEP in the mice. And then I'm going to, at the end, talk about some more mechanistic studies. So most of what I'm showing here are observational studies. We have the mice, and we know they have seizures and sometimes die. And so I record that. And I don't know, I had no preconceived notion of what exactly was going to happen. Now that we have more data, we're starting to generate specific hypotheses. And I'm going to talk about the mechanism that we think produces some of these apneas and kind of talk a little bit about how apnea and how breathing mechanics work in general. And then I'll sum up with a couple slides of where we're going from here to try to understand how the brain causes these negative things to happen and what we might do to, at least in the mice for now, to mitigate that. So the breathing cardiovascular control systems are very important. The main purpose is to deliver oxygen throughout the body. And they work together to do that. The lungs here and the heart here. The heart moves the blood, of course, through the lungs to pick up oxygen, and then through the rest of the tissue deliver the oxygen to it. If one of these systems is totally non-functional, this is detrimental within minutes. So very important. Two very important systems. Excuse me, and two very important systems that are controlled by the brain. The breathing is 100% dependent on the brain. If you have no brain activity, you will not generate your own breathing motions. It's just the way it is. You only need a certain part of your brain stem to do this. So some people who are brain dead in the cortex can still have their own breathing motions. But still, you need that brain activity from your brainstem to do this. The heart can function on its own, but it is highly modulated by the brain, particularly the brainstem. So the way we record these systems in mice is through, at least in our lab, is through headsets that have electrodes in them. So for EEG, the electrodes are implanted in or on the brain. In a sterile surgery environment, and the mice are recovered. In the same surgery, we can also extend ECG leads under the skin to record electrocardiogram. Here, looks something like this on the right. And then breathing is the one that's probably less familiar to anybody, including many scientists, is through what we call whole body plethysmography. And so the way this works is that the mice are in a chamber that's fairly small, that's sealed, except for a metered and controlled airflow in and out to give them fresh air. And then a pressure transducer is connected to the chamber so that we can see the small changes in pressure as the mouse is breathing. As you can see that here in blue. So what does that look like? It looks something like this. The red is the ECG. I'm going to keep this color scheme going throughout. Sometimes the position moves a little bit, but it's always the same color. So the ECG is here. These vertical lines represent individual heartbeats. And if we expand it, we can see that these vertical lines are QRS complexes. That's the big spike you see in an ECG of a human also. It looks a little bit different, which is partially how I placed them in mice, and also partially that mice have a slightly different heart physiology. For the most part, it's the same as humans, 4 chambers, and all the same kind of anatomy. But like a major difference to think about is that humans have heart rates at rest from somewhere between 60 and 90 beats per minute, at least for adults. And mice, adult mice, it's more like 600 beats per minute. So their waveforms have to be squished down to get the signals in. Also, the mouse's heart is very small, so the traveling down axons and things like that is much shorter, and so you have different looking electrical signals. Through our ECG, we also pick up some muscle activity. We have electrodes at different places in the mouse's body, and in between, you're picking up all the electrical activity between there, and that includes muscle activity, particularly during seizures when there's a lot of muscle activity. And at first, I found that annoying because it seemed to get in the way of the cardiac activity, but it actually gives us a very clear picture of what's going on during different phases of the seizure. So it's also useful. The breathing signal is here in blue. Downward deflections are inspirations, are breathing in, and deflections above this line are expirations or exhaling. So we can count breathing frequency. We can see things like this is what I would call an augmented breath, or is a sigh or a yawn, which mice do when just like humans do. And when there's apnea, we'll see a flat line down the middle, which we'll see during some seizures. Okay, so we have these signals here. I play these videos in the software we record them from, and then record the screen. So this signal hasn't filtered quite as much. It isn't as pretty, but it's a pretty good recording. The downward deflections again are breathing in red, these vertical lines of heartbeats, and then we have the EEG. And the mouse that's going to have the seizure is here in the red box. I record from, I'm actually cropping some of the mice that I'm recording from in this video. So we record from several at a time because they don't have seizures that often. So to increase our throughput, we record from many, and this mouse is going to have a seizure when I hit play. And so what I want to look for is that in the second half of the seizure, you're going to see they're going to go into the tonic phase and have tonic contraction of their muscles. And then we're going to see there's an apnea at the same time. Okay, so it's playing. In just a moment, the seizure will begin. Right about now, you can see some activity in the EEG, and you can see the mouse is convulsing over here. The first half of the seizure, it's some kind of clonic convulsions and some wild running and jumping. And during this time, it would appear the mouse is actually breathing at an elevated rate. Heart rate is up. And just about now, they're going to go into the tonic phase. They're still, but there's a lot of muscle contraction. You can see it here in the ECG leads, just a lot of muscle activity. And during that time, we now have a flat signal for our plethysmography. So there's no breathing. Heart rate's still going, although it's kind of slow. And then towards the end of the seizure, towards the end of the tonic phase, we can see breathing resumes. So we have these large gasps at first of breathing, which eventually will transition into a kind of normal breathing that's a bit elevated, probably because the mouse is hypoxic and needs to make up for the loss of oxygen during the seizure when they had so much motor activity and apnea. Okay. So if we compress all of that here, this is what the data looks like. And the thing to note is that the second half of the seizure here, we have this tonic phase, and that corresponds pretty well to when there's no breathing. So if we can see that clearly here, lots of individual breaths and then no breathing here. So this mouse survived the seizure. Some of these seizures are fatal. And in that case, it looks almost identical, except that the breathing after the seizure doesn't occur. And so it just stays apneic and the heart slows until it stops. We recorded in this set of experiments from 8 mice, we recorded 52 seizures. 29 were clonic and 23 were tonic. So the EEG looked very similar, but they didn't have this tonic phase and they didn't have apnea. In fact, breathing tended to increase during a clonic seizure. Of the tonic, many were not fatal, but some were. So we have 8 fatal seizures here and no clonic seizures were fatal. So we believe that this tonic seizure is very important and probably due to the apnea that occurs. And if that's true, then if we could recover this breathing through mechanical ventilation, we may prevent the fatality. And so what we did is we used a different, this is a different mouse model than that experiment. And this mouse is still an SCN8A version. And they have audiogenic seizures. And at this particular age time point, they usually are fatal. And so what we're going to do, I'm going to show you side by side, is one mouse will receive a ventilation where I use a little bulb that I can put snugly over the mouth and force air into the lungs. And the other mouse will receive a sham ventilation. So I'll handle the mouse, but I won't actually ventilate them. And so what we'll see here is we turn the sound on, they have the seizure. This mouse, I begin to ventilate. I kind of view their lungs, their chest, and can see that it's moving. And then this mouse I handled, but I leave to the side. Still the tonic phase here. Eventually you'll see that the muscles relax. The ear goes up. That's how you can tell in a mouse. But breathing doesn't resume. And they do die from this. This mouse, I keep ventilating until I can see that it's breathing on its own, and then I let it go. And, you know, as one would expect, after a seizure, this mouse is fairly out of it. But when you go check the cage the next day, they're in with their brothers and sisters. You couldn't tell which one had a seizure and which one didn't. So it was an effective method. We did this, several times. We took a 10% survival rate and increased it to like 80 or 90% by enacting mechanical ventilation. Okay, so what I've showed you so far is mostly observational, except I guess the mechanical ventilation. But we want to know why does this apnea happen? What is causing it? Of course, if we can understand why apnea is happening, we could potentially come up with therapies to stop it. So to get into what we did next, I have to talk a little bit about the mechanics of breathing. So breathing is a motor activity. In humans, breathing at a minimum requires the motor oscillation of the inspiratory muscle, so an inspiratory oscillation. The main inspiratory muscle is your diaphragm, so it's under your rib cage. And when it contracts, as we can see now, it goes down. That expands your thoracic cavity in your lungs so that air can go into your lungs. Then at rest, your diaphragm relaxes and air just passively goes out. And this is how you breathe. If you exercise, you'll also start to engage expiratory muscles so that you can force in and air in and out quicker. But in a resting individual, this is what you need. You always have diaphragm contraction, rhythmic diaphragm contraction. You have to. This is how all breathing in humans works. There are some animals where expiration is the primary driver, but in humans it's inspiration. And if we looked at the muscle, if we recorded it, it would look something like this. So when the diaphragm contracts, we'd see a bunch of electrical activity from that muscle. And when it relaxes, we would see very little activity. And this repeats forever as long as you're alive and you're ventilating yourself. So we can imagine that if you're not going to breathe, there are kind of two ways this could occur. You could just never have muscle activity. The muscle activity could stop. Or alternatively, you could have muscle activity that occurs constantly. If this never relaxed, it's kind of like an inspiratory breath hold. That's what we call it. So if you go to the pool and you decide to jump in, you breathe in, you hold it by contracting your diaphragm, and you jump in the pool. That's how you hold your breath. And so during a seizure, we could be recruiting parts of the brain that cause this to happen and forcing an inspiratory breath hold for an extended period of time during the tonic phase. That was my hypothesis. Since there's so much motor activity, so many muscles are contracting, why not the diaphragm? It could be contracting too. So to test this idea, we recorded from the diaphragm in mice and then induced audiogenic seizures. So this is yet again another kind of version of how we use the mouse model. In this version, the mice are adults, and these seizures are always survived. So I can instrument a mouse to record and then stimulate multiple seizures to get repetitive results. When I record the activity from a mouse, the diaphragm, we can see that this diaphragm activity, this muscle activity, the bursts line up with inspirations. So these blue are the same plethysmography traces that I've shown you before, and they line up really well with the muscle activity from the diaphragm. In fact, if we get a larger breath, we get a larger muscle contraction that we see here. So then we can take this mouse and induce a seizure by playing one of these 15, I used a very precise tone in this case, 50 kilohertz, that causes a seizure. So before the seizure, we can see this kind of normal breathing pattern, normal muscle contractions. Initially, during the first part of the seizure, we see an elevated breathing like we saw before, and we can see this in the diaphragm, these faster breathing contractions, muscle contractions. But then once we get to the tonic phase, we switch into this tonic contraction. So we just see really a massive amount of muscle activity here. I actually, to be able to see this clearly, I had to expand the trace and then kind of cut it off. So it's actually, this is cut off. There's even more muscle activity. And we stop seeing this nice, even, smooth contraction and relaxation that we see during normal breathing. So this isn't a causal experiment, but the contraction of the diaphragm like this is sufficient to cause apnea. And so I would say that most of this apnea is due to contraction of the diaphragm. There is a period at the end where the diaphragm relaxes and it takes a while for breathing really to get going. There might be a secondary mechanism going on that contributes to that. But since this is a very novel observation, I would say, if you read neurology textbooks and you look up tonic seizures, they will state that breathing muscle contraction is probably causing apnea during tonic seizures. And they won't reference anything. There are no papers. And nobody's measured this in humans. It'd be pretty invasive. And there was really just recently a paper to do this type of recording that came out that I copied. So I was able to do this experiment. And so I'd say this is pretty novel, and we're going to follow this up with a few sets of experiments. So first, what neural mechanisms during a seizure are causing this to happen? So very briefly, your diaphragm is a muscle like any others, and so there are motor neurons in the spinal cord that make these muscles contract, but they're under command from neurons in the brain. And this picture is showing you the primary motor cortex, so right on top. For reasons I'm not going to get into here, I think that these are actually not the ones that are causing the muscle contraction during apnea. And there are several other upper motor neuron pools that could be contributing. So there's the cortex, but there's also these other motor nuclei in the lower part of the brain that also send signals to motor neurons in the spinal cord to cause muscle contraction. And so the experiment here is to record from these different regions and see which ones become active during apnea. Some of these regions a mouse can live without. You can destroy the red nucleus and mice are fine. They have slightly worsened motor behaviors in certain tests, but they survive and they're fine. So if this nucleus was, and there is some reason to believe this innervates muscles that control the diaphragm. So this could be possible. This could be occurring. Excuse me. So we could then intervene once we know which regions of the brain are important. But this first experiment is more observational. Secondarily, there is some neural circuitry in the brainstem that is pretty well described that we can attempt to use to interrupt this apnea. So just as a reminder, we think that this oscillatory motor activity that's normal has become tonic during the tonic phase of a seizure, and that's why they have apnea. So if we can inhibit this inspiratory activity, we could potentially recover this. And so in this set of experiments, we intend to do that. The inspiratory oscillator, the neurons that originate the inspiratory activity are in your brainstem, and they are modulated by several neuronal populations, but one very powerful one is this Botzinger complex, which has a fun story actually for its name, but we'll get into it now. If stimulated, they inhibit inspiration. So we plan to use some newer technology called optogenetics, where we can use laser light to stimulate these neurons and then inhibit this inspiratory activity and kind of effectively we can cause expiration and kind of regain this oscillation again during the seizure and recover the breathing. This would be very exciting to do as a rescue, and also it would confirm our hypothesis that there's this seizure activity has spread to the inspiratory oscillator and is causing this apnea. Secondarily, as I pointed out before, there's sort of this gap after the tonic phase where there still is no breathing, which we can't really ascribe to this tonic motor activity. So perhaps at this time point, the inspiratory oscillator is not receiving enough drive. The inspiratory oscillator requires, based on experiments in the field, it requires constant input to function. So maybe it's not receiving enough input of it post-ictally because the brain is kind of suppressed. One neuronal region that has a very powerful, can have a very powerful influence on this inspiratory oscillator is called the RTN, which is a neuronal group that I've worked on extensively. And we can stimulate this with optogenetics also and see if we can recover breathing after the seizure. So we're kind of looking to use these different neuronal groups to manipulate the circuitry here and try to rescue this apnea during and after a seizure. And so those are our future directions. We're hoping that this and what I showed you in that previous slide, we'll be doing these experiments in the coming months to year, hopefully maybe even by next December, we could even have some results to show you. I certainly hope so. And so with that, once again, thank you. I'm sorry it didn't work out yesterday. Please contact me with any questions, and I will be happy to see all of you in December. Look forward to it. Everybody be safe and have a happy